So let's go ahead and get started. Um, I'm just going to briefly introduce the next several sessions that we have on the agenda, which I consider to some degree the heart of the discussions. But before I do that, I would just like to acknowledge the Borough's Welcome Fund, who actually were the ones that planted the seed for this particular workshop. Borough's Welcome has been a organization that has done a lot of work in the area of regulatory science, and I think importantly have been very interested in supporting through funding the area of regulatory science and developing it as a discipline. And so I'd certainly like to acknowledge them, as well as the National Academies, for their ongoing interest in topics related to regulatory science. I'm now am the acting commissioner of FDA. I've been in that role now for several months. And I've said often that for a long period of time, FDA was viewed as a regulatory agency, and a regulatory agency that does a little bit of science. And I think that certainly Peggy Hamburg, my predecessor, as well as myself, feel very strongly that FDA should be more appropriately viewed as a public health agency that makes science-based regulatory decisions. And in order to be able to do that, you have to have very sound and very strong regulatory science. When we stand on sound scientific grounds, we make very good regulatory decisions. And when we don't have that solid science, then that's where we can tend to get into some difficulties. And I guess one of the themes that I hear for a workshop like this is, to what degree can we use science that's somewhat outside of our comfort zone to make regulatory decisions? And so in the next several sessions, you're going to hear a couple of topical areas in which I think we're in that space. And so the question and the issue of how do we use some of that science in our regulatory decision making is quite profound. And the first of those is in the biomarker space. This afternoon we'll be talking about clinical trials, and you heard some of that in the first session. And then the third area is in post-marketing surveillance and pharmacovigilance, and they're all very critical, and they're all areas where the science and the information integration is really taking off. And so with that, I'm going to turn it over to John Wagner, who's going to moderate the first session.
Thanks, and pleasure to be here. So this first deep dive session is about biomarkers, which of course is really very cool because of course for regulatory science the biomarker connection is really quite long and deep. So ever since critical path, actually well before that, the NIH FDA consensus conference back in 1999, there's been this history of pushing the envelope with biomarkers. And yet we haven't made enough progress, not nearly enough progress. And today, in the vein of this sort of future-directed kind of talks, we're gonna dive a little bit more deep there. And I think that you all know that the world of biomarkers is totally heating up right now, whether it's 21st Century Cures, the Biomarkers Consortium, the FDA Biomarker Qualification Pathway, and a survey that we'll hear about, CPATH, Brookings FTA workshops. The field right now is just exploding with activity. The question is whether we can pull it together and do something worthwhile. So without further ado, I want to ask MARC SALIT to come up. He's a leader of genomic scale measurements at the National Institute of Standards and Technology, and he's going to provide kind of context and tone setting here and probably push the envelope a little bit.
Thanks so much, John, and thanks so much to the organizers. I lead a group called Genome Scale Measurements at the National Institute of Standards and Technology, NIST, and we're part of the U.S. Department of Commerce. think about the dissemination of the base units, the meter, the kilogram, the second, the candela, if you want to get a little more esoteric, the ohm, the watt. I'm an analytical chemist and atomic spectroscopist by training, and so I've been, I got bored and decided to see if what we had built as a framework for doing rigorous metrology, understanding the measurement science behind the results we were getting, as well as understanding sources of variability and their relative magnitudes, understanding when we're seeing artifact and when we're seeing something authentic in our signals. If we could take what we'd learned there and start to apply it into a different space entirely, and that is measurements of multiple cohorts of biomolecules. So I've been working at this. I wasn't doing work in the radiographic measurement of gene expression, but rather I started in when we were looking at DNA microarrays and looking at highly multiplexed data and asking what are the, at some point, and there's talk about esoteric, metrology has been discussed as the morality of measurement. So could we assess from a moral basis the validity of measurement results? Let's go to the slide here. So I think about this as a three-legged stool. There are sort of three principles. There's metrological traceability, there is measurement uncertainty, and there is method validation. And on top of those three legs sits confidence. So when you can understand where did I get the units on my result, what is the likely dispersion around that result, and what evidence have I built in a systematic way to establish that I'm actually reporting a result that has been rigorously considered. From that, and I'm not saying exactly what you need to be traceable to, and I'm not saying just how uncertain things are, nor am I saying that my validation evidence is comprehensive, but rather when you can say something informative around those principles, you can start to understand how confident you can or can't be in your measurement. And this has been really essential, I think, in moving the needle on biomarkers and on biomarker. A lot of my biomarker experience has been working collaboratively in an NCI program, the National Cancer Institute's Early Detection Research Network, which I'll be careful not to be disparaging about it, but it's been a biomarker hunting network where people are looking for early indications that would inform therapeutic decision-making for cancers and broadly writ. And it's not been a super efficient network at generating new validatable FDA-approved biomarkers. While it's got a handful, there's been lots of blind alleys. And so a lot of this comes from, and I've got a more detailed slide here on traceability, a lot of this comes from the lack of the ability of one group to repeat the results of another group. So the repeatability problem is nothing new, though it's quite prominently recognized now, starting really with the Begley piece from 2012. So a lot of the problem is you can't compare results from one study to another because they're not traceable to the same artifact, or they don't have the same units, as we heard in one of the remarks from the floor on platelet measurements. So traceability isn't that fancy, and while my colleagues in the National Measurement Institutes around the world will, it's our full-time job to some degree, metrological traceability is really just how did I get the units on this answer? Often we achieve in chemical measurements, physical measurements, we often achieve traceability through calibration, conversion of the volts that you read out of your analog to digital converter on your instrument into a unit that's of interest in your study. In biology experiments, particularly in pathology experiments, we end up looking for the enrichment of a molecular signal between a cohort of control samples and a cohort of case samples. And that enrichment is, in fact, what we would hope to reproduce. And it's got feet of clay because of the challenge of repeating the control cohort. Again, the panel remarked a little earlier about retrospective assessment of, and Russ made the comment about retrospective assessment of large sets of data from electronic health records and curating out well-matched controls and study cases. Those are the kinds of things that hold a lot of promise for robust identification of biomarkers. Measurement uncertainty is a pretty, I'm not going to spend a lot of time on this. It's how do I put the plus minus? How do I put the error bars on the value? And there's a science of doing that of its own. I will say, here's where I'll say that doing this for genomics is really challenging. Doing traceability for genomics is really challenging. The very concept of actual variant, genomic variant measurement or determination doesn't fit in this framework. All this breaks. So I've got myself a full-time, long-term job leading a group called Genome Scale Measurements because we're trying to develop the metrology of not only, gee, what's a gene expression signature traceable to, and what's my measurement uncertainty on a gene expression signature, but is this SNP in this location an A, a C, or a T, and that's plus and minus what? And it's traceable to the nucleotide. Anyway, so we're trying to formulate this in a robust way, the metrology of biological signatures and the metrology of nominal properties or sequence and sequence variants in genomics. This has a lot of overlap with what Elizabeth and Jim were both talking about, and ultimately the seat of that stool feeds into the decision-making process that goes forward in a study, both for drug qualification and for surveillance thereafter, for decision-making in general, hypothesis support or rejection. Method validation, as I said, we got pretty good at this in chemical analysis, and that is systematic ways to build an objective case that the measurement result I'm reporting is not an artifact, but rather is a robust estimate with a trustworthy measurement uncertainty band around that. And this is all around analytic validation, not clinical validation. And I would posit that analytic validation, making sure that what you're actually measuring, as we heard a little nod to a few moments ago, is in fact a key piece for going on. So now I can make decisions when I've got this stuff lined up is really the piece there. I want to talk about ways we do this really quickly. The target plot should be familiar to most in the room. One of the things we do is we take that idea of a target plot and bring it to multi-lab or inter-laboratory studies. And I've always liked this representation where in a world where you've got variance increasing on this axis and the proportion of outliers increasing on that axis, these are the kinds of results from inter-lab studies that you might observe, and you can really start to classify, wait a minute, is my community or my population of labs harmonious, or am I out of luck? And here's some results from a microRNA study where we've got 3 different techniques. We've got a hybridization technique, a next-gen sequencing technique, and PCR methods. And you can look at results, and this is for 5 microRNAs because the PCRs are kind of one at a time, so we don't have a great population. And you can start to see if we look at ratios, and again, these are traceable to a control in these experiments. There are sort of 3 different things we're measuring, 3 different ratios, and they go across, and each lab is one of the points, and there are tick marks for the labs. And you do this, and you can repeat this for a second round, and you can repeat this for a third round, and so on. And again, try and this is being done under the auspices of that early detection research network, and this is being done in order to evaluate are we chasing artifacts when we're measuring microRNAs as biomarkers? And I'll quickly finish because my yellow light is really on. Some work we've been doing in genomics for gene expression has been trying to use spiking controls in clinical samples of interest and going ahead to try and can we use spiking controls and tell a good experiment from a bad one? So we've got 4 measures here, a familiar rock curve I'll call your attention to in the upper right. And here's a good lab, and here's a not so good lab. Same samples, same study, and now we've got a way just from the spiking controls to tell them apart. And that's the kind of stuff we're trying to build in to give us a more rigorous framework for biomarker, to give us a more robust biomarker pipeline. I'll put it that way, and I'll stop now, 49 seconds.
Great, thanks. So we're